Hi everybody. Today we're going to talk about some of the ecological and evolutionary consequences of viral infections. I really like this figure from a great review by Maya Breitbart. If you want to learn anything about viral ecology, I highly recommend looking at Maya Breitbart's publications. In this particular figure from this review from 2012, she shows these cartoons that are showing three examples of evolutionary consequences of viral infections. I'm going to go over each one in particular, but quickly the three are kill the winner, that is the most abundant members of the population are more likely to die from a viral infection. The second one is enslavement, which means that the viruses can actually put their hosts under their own control. Third one is gene exchange, which we've talked about already. Viruses are a really important vector for genetic exchange among species of microbes. All right, talking about kill the winner. First, this is a fundamental concept of microbial ecology, which is that the more abundant you are, the more susceptible you are to a viral infection. Because viral infections are more likely when members of that population are very densely populated, when they are very close to each other physically. As we are all experiencing right now, we are susceptible as humans to viral infections when we are living in very close proximity to each other. That is also true for all other species on the planet, including microbes who live in the ocean or microbes who live in soil or anything else. So this diagram is showing an example of we have some kind of diverse microbial community. These green guys all of a sudden take off. They have a bloom of their population. Good for them, great. But because they are so dense, they are now susceptible to a viral infection. So now we have these green viruses infecting the green cells. The green cells get beaten back down by the viruses to a point where their population is now back to where it was at the beginning. Now this is drawn to be very similar but slightly different to the original state to highlight the fact that it doesn't necessarily go back to exactly the way it was before. Of course, biology is never so simple. Sometimes it's a little bit different than it was before, but the idea is that there's some kind of return to equilibrium, and that overall the net effect of the consequence that more abundant populations are also more susceptible to viral attacks is that viruses kind of keep a check on the ecosystem from getting totally out of control. This is one of the important reasons why most ecosystems are not dominated by one single species. We generally think of that as an unhealthy ecosystem when there's one species dominating to the detriment of everything else. And one reason why that doesn't happen very often is because of this effect of viruses killing the winner. Okay. One example of this, Emiliania huxleyi. This is a species of coccolithophore, which is a type of phytoplankton. We'll talk about phytoplankton more in the next week of lectures, but briefly right now, this is a type of eukaryotic microbe. It's a single cell, but it's a eukaryote. You can classify it as a type of algae, although as we'll see next week, algae is not a terribly useful taxonomic term, but it's a microbe that does photosynthesis in the ocean. It can be really abundant in some places. Their blooms can be super, super dense. It can cause the nutrients in the ocean to be depleted because they are so abundant. And they're adapted to those points where there's low nutrients as well. They look like this. They have this armor of calcite shells, so they're covered in chalk, essentially. And because of that, they play really important roles in the carbon cycle and also the sulfur cycle, which can affect things like cloud formation. So these guys, even though they're kind of weird, they're carbonate-covered algae. They are really important players in multiple aspects of global ecology. They are infected by viruses, as pretty much everything on the planet is. Here is a satellite image of a coccolithophore bloom off the coast of Alaska. We all know that Alaska is very big, so you can appreciate how large this bloom is. And the milky white color is probably due to the fact that this coccolithophore bloom was being subject to a viral infection. because that's what happens when you have a population bloom. You are almost immediately followed by a viral infection. So the milky white color is probably due to viruses lysing the cells, and then those carbonate shells being leaked out into the water and starting to dissolve to form this milky white color. If you have a big bloom of coccolithophores that are sinking down to the sediment, the bottom of the ocean, and you do that for thousands or millions of years, and then those rocks with those dead coccolithophores are uplifted onto the coast where people can see them, then you get something like the White Cliffs of Dover. The White Cliffs of Dover, this famous geological formation in southern England, this is from dead coccolithophores. These carbonate minerals are from these carbonate-covered phytoplankton that died and sunk to the bottom of the ocean. So this is another way of showing just how abundant these coccolithophore populations can become. So here is a picture of the virus that was discovered relatively recently that infects Emiliania huxleyi. For a long time, it was kind of assumed that the carbonate shells of Emiliania huxleyi were adaptations to prevent viral infections, that the viruses couldn't get through that armor. But then this was discovered, and in fact they are infected by their own virus. Here it is. And this slide is showing kind of formally the ecological consequence of big viral infections. Again, next week we're going to talk about the marine carbon cycle in particular, but really quickly right now, the traditional marine carbon cycle is that you had phytoplankton doing photosynthesis that were eaten by small grazers like krill and other small crustaceans, which were then eaten by carnivores like large fish. When this organic matter dies and sinks, it becomes particulate organic matter or dissolved organic matter. That's POM and DOM. And that sinks down to the bottom of the ocean, and then you have heterotrophic bacteria that then decomposes that biomass to make CO2. So that's kind of the cycle where CO2 is fixed by the phytoplankton, and at the other end you get bacteria decomposing that biomass to make CO2 again. When the importance of viral infections in the marine carbon cycle was appreciated, it was realized that viruses kind of act as a shunt that bypass one or more steps of the carbon cycle by immediately converting the phytoplankton biomass into dissolved organic matter by lysing the cells and leaking the contents of those cells out into the ocean. So it immediately goes to DOM, which can very quickly go to CO2 by the actions of heterotrophic bacteria. So you can recycle the carbon that was fixed by the phytoplankton back to CO2 relatively quickly, all in the surface of the ocean. So the CO2 comes in and then it goes back out again due to viral infections. And it doesn't require this complicated food web to release the CO2 again. Okay, we'll talk about that again next week, but for now, here is the viral role in that cycle. Here are some numbers to talk about just how globally important viral infections in the ocean can be. Again, we've talked about before how there are an incomprehensible number of viral infections in the ocean every second or every day. It's estimated that this kills 20 to 40% of all marine bacteria every day, which releases billions of tons of carbon every day. As shown here, this is another version of what I showed on the previous slide, where you've got phytoplankton fixing CO2. When they're infected by viruses, the cells break apart. The contents of those cells leak out to become particulate organic carbon or dissolved organic carbon, the only difference between these two being whether the carbon is in particles that can be sticky and clumped together, or it's very small molecules that are dissolved in the water. Either way, those get released back as CO2 relatively quickly, thanks to bacteria, and released back into the atmosphere. So only really addition on this slide compared to the previous slide is putting some numbers on how many gigatons of carbon we're talking about per day, and it's a lot. Okay, that was the kill the winner concept of one of the three main evolutionary consequences of viral infection. Second one I want to talk about is enslavement, where the viruses actually affect the metabolism of the host in a way that is more complicated than simply killing it. So this is best explained with an example, but before I mention that example, I want to talk about a little bit of viral biology to give you some background information. So here's the classic scenario where the virus lands, injects its genetic material into the host. The host is then forced to make copies of the virus, which then multiply and break out of the cell. Sometimes when this happens, though, the viral genome actually gets incorporated into the host genome. So this is what we call a lysogen, when instead of immediately triggering an infection, the viral genome instead gets incorporated into the host genome. Then we call it a lysogenic virus. And because this frequently happens during viral infections, and then at some point something triggers the prophage, it is now called, when the viral genome is inside the host genome. If you trigger the prophage to become active and then trigger an infection, then by accident, when that viral genome escapes the host genome, it can take with it some of the host genes. Okay, so this is kind of the canonical example of how viruses end up transferring host genes among hosts, is by an accident of replicating their DNA. After being incorporated into the host genome, they grab some of those host genes and take them along with them at the next infection. This is not the only way this can happen, but it's kind of the textbook example of how this can happen. Okay, so because this is a thing that happens with viruses, they sometimes steal genes from their hosts. Not only can they just accidentally transfer those genes to the next host, or the host after that, but you can also get really complicated things like this, where the virus can actually trigger expression of those genes during viral infection. Okay, so this is a slightly complicated example. Let me take some time to explain it. Here we have a cyanobacteria, one of the members of the phytoplankton in the ocean. It's doing photosynthesis to fix CO2. And one response that cyanobacteria have, Prochlorococcus is the genus of cyanobacteria we're talking about. One response they have to a viral infection is to basically shut down their metabolism. So that doesn't do the particular cell that was infected by the virus any good. It's probably going to die anyway, but by shutting down its own metabolism, it can stop the viral infection in its tracks and prevent all of its brothers and sisters from being infected after it is killed by the virus. So that's a really common response to viral infections, to shut down metabolism to stop the infection where it is. So this particular virus that infects these cyanobacteria, it encodes multiple genes of photosynthesis in its own viral genome, which it can also trigger the expression of. So it can actually make photosynthesis keep going. It can maintain photosynthetic activity in the host to counteract the stress response of shutting down metabolism. So by keeping the machinery of photosynthesis going during the infection, it can prevent the host from shutting down the viral infection. The viral infection can proceed. It can make copies of itself by using the energy generated during photosynthesis. So in this case, it's not the virus is not encoding photosynthesis genes just to help the host because it wants to help it do photosynthesis. It's helping the host do photosynthesis so that it can better kill it and infect it and make more copies of itself. Pretty insidious. This is a paper providing really strong evidence for this story. So I've already presented the conclusion to you on the previous slide. I just wanted to show this figure as an example of how they figured this out. Okay, so here is a phylogenetic tree. Again, wanted to give you another example of the power of phylogenetics. And all of the green ones are photosynthesis genes that are encoded by the cyanobacteria themselves. Here are freshwater cyanobacteria. Here's a different genus of cyanobacteria. And then the red ones are viruses, are photosynthesis genes that were encoded in viral genomes. So in this example, the red viral sequences of these particular photosynthesis genes are phylogenetically descended from their hosts. So this is a Prochlorococcus virus. and those genes are descended phylogenetically from the Prochlorococcus host. A second example of a Prochlorococcus virus encodes photosynthesis genes that are directly descended from its Prochlorococcus host. Also with the Synechococcus, which is another genus of cyanobacteria, it has a virus that has photosynthesis genes that are descended from the photosynthesis genes from its host. So really clear evidence that these genes came from their hosts, and then the viruses encode them and use them against their hosts to maintain metabolism during the infection. I think that's a really cool story. I hope you liked it too. Third consequence of viral infection is vehicle for gene exchange. We've already mentioned this in this lecture and also in previous lectures. I used this slide before to talk about transduction. That's the technical term for transfer of DNA due to viral infection, and mentioned these crazy large numbers about how many viral infections there are per day. And even though I already mentioned it, here's another example, perhaps of an example of a virus transferring genes between hosts in action, or maybe it's an example of the previous one with the viruses encoding photosynthesis genes. I think it's not perhaps clear yet in this example. So what is this? This is another example of phytoplankton. Here's a green algae. In this case, instead of cyanobacteria that's being infected by a virus, here's one green algal cell with multiple viruses inside it. So the cell is not long for this world. It has all these viruses replicating inside of it. And these viral genomes encode genes that are used in fermentation. So the idea is that the viruses might enable the host to grow and therefore make copies of the virus in anoxic environments by enabling the host to do fermentation. You don't normally think of algae doing fermentation because they're photosynthetic. So maybe that becomes a benefit to the host, that it can adapt to new conditions. And over time, it's possible that these genes could be transferred to the host genome. And then the host would always have the ability to do fermentation and survive anoxic environments without a viral infection. So that is one potential direction the story could go. But I think that probably the interpretation for what's happening at the moment with this relationship is that the host is using this to manipulate the metabolism of the virus is using this to manipulate the metabolism of the host to probably as a counter response to a stress response that the host is mounting in response to the viral infection, perhaps by shutting down metabolism. And so the response to that is that the virus encodes these genes for fermentation to generate some metabolic energy to power its own viral replication inside the host. I think that's probably the more likely scenario, in which case it's another example of viral enslavement. But you can imagine how over time this could also result in transfer of these genes into the host. Okay, so those were really quickly some examples of the many different consequences that a viral infection could have. And at the end of this lecture, I just briefly want to mention that the hosts are not completely defenseless against viruses. Over the billions of years of microbial evolution, bacteria and archaea and microbial eukaryotes have evolved various mechanisms for defending themselves against viruses. One general category of strategy to deal with viral infections is to have an immune system. We normally think of immune systems as something that animals have, or at least multicellular animals have immune systems. But bacteria and archaea and microbial eukaryotes also have their own immune responses. It's an ancient concept to have some kind of system to deal with a potential viral infection. And one type of immune system, especially in bacteria and archaea, is CRISPR. So CRISPR stands for clustered regularly interspaced palindromic repeats. You may have heard of CRISPR before, just like in popular culture, reading the newspaper, because it has now been used as a genome editing tool. And it is receiving a lot of attention for its power to, for example, edit the human genome. And it's also a really powerful tool in research laboratories to just understand how organisms work. And to do genetic engineering. And so that's probably how you've encountered it before. But today I want to mention that CRISPR, in fact, is an immune system that bacteria and archaea use to defend themselves against viral infections. This is how it was discovered is by studying how bacteria defend themselves against viruses. Bacteria and archaea have been using CRISPR systems for probably billions of years to mount responses to viral infections. And it's only in the past few years that humans have discovered that it exists and have figured out how to turn it into a genetic engineering tool. Okay, so what is it exactly? I want to spend a little bit of time to show you the natural ingenuity of how CRISPR works. So it's called clustered regularly interspaced palindromic repeats because a CRISPR locus is composed of alternating regions of DNA that either are a repeat or are a spacer. So repeat is the same sequence that happens over and over and over again. So each one of these diamonds is exactly the same sequence that shows up over and over and over again. And then a spacer are the regions in between those repeats, and those are variable. So that's why they're shown here as different colors and also numbered at the same time. And the way this works is that a virus infects, as shown in this slide. Virus infects the host. Host recognizes the viral infection, synthesizes a molecule, an RNA molecule that matches the viral DNA, and then inserts a segment of that RNA into its own genome. It turns it into DNA so that it is now part of the genome. So the host has actually synthesized a molecule that matches the viral genome and then stored that sequence in its own genome. So you can already see how this turned into a genetic engineering tool, because it's basically a tool that bacteria and archaea use to edit their own genomes. And they edit their own genomes so that they have a library of past infections. So they have a record of which viruses have tried to infect them in the past, so that if that virus tries to infect them again, they can immediately recognize, hey, I know you, and that allows them to mount a very quick response against that virus. It's, in concept, works like all other immune systems. There's a memory component of remembering who has infected you before, and so that if that virus tries to infect you again, you can very quickly identify it and mount a response against it. And so when you look at bacterial and archaeal genomes, going back to this slide, you can read off little snippets of viral DNA sequence that have tried to infect that host before. And not only that, but they are inserted in order so that you can read off in order from ancient to more recent which viruses have attempted to infect that host and in which order. Converting the order into an actual amount of years is tricky, and that's something that people are still working on, but it has been shown that they do occur in order. It's quite possible that there's some kind of a memory wiping function that over time the genomes probably get rid of spacers matching viral DNA that were so long ago that they're no longer useful. There are probably some kind of selection component to this that are only selecting the regions that are actually useful for defending the host against viral infections. But that's all kind of brand new research, and there's much less research going into how this actually works in bacteria and archaea compared to all the billions or trillions of dollars that are going into how to use this as a tool for genetic engineering. But I wanted to mention to you, make sure that you knew that this is a real thing that is used in the wild every second of the day by bacteria and archaea to protect themselves against viruses. So just to end with acknowledging that this was a Nobel Prize in 2020 to Jennifer Doudna and Emmanuel Charpentier, who came up with some really critical innovations and discoveries and inventions that allowed this natural immune system and microbes to be turned into a human genetic engineering tool. And I have a little snippet of their paper here that shows how they recognized very early on how this fundamental concept of the bacterial and archaeal immune response could be turned into a very specific gene editing tool. Okay, there are a million resources online to learn more about CRISPR. If you like, I encourage you to do that. And that's all for today. See you next time.